Hello, welcome to my poster. My name is Zhijian Li, and today I will introduce you a new computational method to estimate chromatin accessibility from single-cell ATAC-seq data. So first of all, what is ATAC-seq? Basically, ATAC-seq is a protocol to profile chromatin accessibility in a genome-wide manner. And this protocol is very sensitive and straightforward. Very recently, people also combine ATAC-seq and single-cell sequencing as single-cell ATAC-seq. And now this protocol is commercialized by 10x Genomics. Usually, to be able to analyze single-cell ATAC-seq data, the first step is to detect the peaks by combining our cells. And those peaks will be used as features to create a count matrix for downstream analysis, such as cell clustering and visualization. However, it's very challenging to analyze this count matrix because first, this matrix is highly dimensional, and so usually the number of peaks is more than 100,000. The second problem is this matrix is binary because for each single cell, the chromatin in a particular region can be either be open or closed. Finally, this matrix is very sparse, and since there are only two DNA copies in each single cell, and also when you do the experiments, usually you will have some loss of DNA material. So this leading to dropout events. Here, to address these problems, we develop a computational method called SOpen to estimate the chromatin accessibility of each single cell for each peak. The theory behind SOpen is we can try to impute this binary matrix using a machine learning model called regularized non-negative matrix factorization. The output is another matrix where each element represents a square of a peak in a cell. Also, SOpen can also provide a low-dimensional matrix. So the output can be used for downstream analysis. We first test if SOpen can improve the clustering of single-cell ATAC data. To do so, we collected for publicly available datasets, and we also compared SOpen with a number of different imputation methods. So you can see here, we evaluated the clustering results. So you can see here, SOpen outperforms the computing method by providing either the highest or the second highest clustering results. Next, we would like to apply SOpen to study the earlier regulatory changes in kidney fibrosis. To do so, we generate single-cell ATAC data from intact mouse kidney at different time points after injury with fibrosis. So our experiments recovered more than 31,000 cells, and we applied SOpen to generate the low-dimensional matrix. We also performed data integration with Harmony algorithm, and finally we cluster and annotated the cells. Notably, we are not only able to identify all the major cell types in the kidney, but also some rare cell types, such as podocytes. Moreover, we also identified a new cell types which we termed as injuring proximal tubular cells. Finally, we compute the cell proportion across different time points, so it's quite clear that some cell types are increasing, like macrophages, injuring proximal tubular cells, and also there are some cell types which are decreasing along this process. So we next used hint attack to compare transcription factor activity between some cell types, and we found that the enduring proximal tubular cells have overall low factor activity. A key processing in kidney injury is fibrosis, which is caused by differentiation of fibroblasts and parasites to myofibroblasts. So here, to dissect the potential differentiation trajectories, we performed a differential map embedding of the fibroblast. And this analysis identified three major branches as formed by fibroblasts, parasites, and myofibroblasts. Next, we build a cellular trajectory across the differentiation from fibroblasts to myofibroblasts. And then we calculate the correlation between the TF gene accessibility and the bonding activity along the trajectory, and we identified a number of factors which are relevant for this process. One example is RUNX1. So on the right-hand side, we showed the ataxic footprints of RUNX1. So we can observe that RUNX1 has a deeper footprint in the late stage of this differentiation process. And this indicates that RUNX1 is an important transcription factor for myofibroblasts. To validate the function of RUNX1 in myofibroblasts, here we perform immune staining and quantification of RUNX1 signal intensity in transgenic PGF-beta mice that genetically target fibroblasts and myofibroblasts. So here on the bottom, we can see that the RUNX1 is colocalized with TGF-beta signaling. To summarize, S-Open is a computation method designed to address the sparsity and high dimensionality of single-cell ataxic data. We showed that S-Open improved single-cell ataxic clustering, and we also show S-Open can be used to study regulatory dynamics for complex datasets. And finally, our analysis identified RUNX1 as a relevant factor for myofibroblast differentiation. With that, I want to end my poster and want to thank you for your attention.